Now just quickly compare these two sessions of WBC counting and RBC counting. So here we could say calculations WBC total count. So number of WBCs in first field is found to be 56. Number of WBCs in second field is found to be 48. Number of WBCs in third field is found to be 61. Along with that, number of WBCs in fourth field is found to be 51. So total number of cells counted is turned out to be 216. It is represented with the help of alphabet N, capital N. And area counted 4 into 1 mm square. 4 mm square. Then dilution factor 1 by 20. Depth factor 1 by 10. So WBC cubic mm of blood, N by dilution factor and depth factor into area counted. So N into 200 by 4, N into 50. We could see the result. Okay, like that. There's some small differences are there in case of RBC. Volume of one small square is found to be 1 by 4000 mm cube, not mm square, mm cube. Volume of 80 smallest squares, 80 into 1 by 4000 is equal to 1 by 50 mm cube. 1 by 50 mm cube. If the number of cells in 1 by 50 mm cube is x, the number of cells in 1 mm cube is equal to x into 51. The dilution of blood in 1 is 200. The number of RBCs in 1 mm cube in diluted blood is equal to x into 50 into 200. X is found to be 526. Okay. So 526 into 50 into 200. Volume of one smallest square 1 by 4000 mm cube. Volume of 80 smallest square 80 into 1 by 4000 is equal to 1 by 50 mm cube. If the number of cells in 1 by 50 mm cube is x, the number of cells in 1 mm cube is equal to x into 50. So the dilution of blood in 1 is 200. The number of RBCs in 1 mm cube in diluted blood is equal to x into 50 into 200. X into 50 into 200. Next experiment, estimation of protein by Lowry's method. Lowry's method is a very important method used for protein estimation. Protein estimation in the sense qualitative estimation to determine the amount of protein. So quantitative amount of protein in the given sample. Materials used: test tube, pipette, glass rod, colorimeter or spectrophotometer. So reagents: 2.63% sodium potassium tartarate solution, 1% copper sulfate solution, 2% sodium carbonate in 0.1 NaOH solution, protein standard solutions. Once again I am going to read this. 2.63% sodium potassium tartarate solution, 2.63% sodium potassium tartarate solution, 1% copper sulfate solution, 2% sodium carbonate in 0.1 normal NaOH solution, 2% sodium carbonate, 2.63% sodium potassium tartrate solution, 1% copper sulfate solution, 2% sodium carbonate in 0.1 NaOH solution, 2% sodium carbonate in 0.1 normal NaOH solution, protein standard solution, dissolve 10 mg of bovine serum albumin or BSA in 100 ml, 0.1 normal NaOH solution, keep frozen. And working solution, mix 100 ml of sodium carbonate solution with 1 ml sodium potassium tartrate solution and 1 ml copper sulfate solution. Once again I am going to repeat this. 2.63% sodium potassium tartrate solution, we need that. 2.63% of sodium potassium tartrate solution, 1% copper sulfate solution, 2% sodium carbonate in 0.1 normal NaOH solution, 2% sodium carbonate, 2% sodium carbonate in 0.1 normal NaOH solution. Protein standard solution, dissolve 10 mg of bovine serum albumin or BSA in 100 ml, 0.1 normal NaOH solution. Keep frozen. Then working solution, mix 100 ml of sodium carbonate solution with 1 ml of sodium potassium tartrate solution and 1 ml copper sulfate solution, that is acting as a working solution. 100 ml of sodium carbonate solution with 1 ml sodium potassium tartrate solution and 1 ml of copper sulfate solution. We just need 1 ml, but still we are preparing 100 ml of this copper sulfate solution to get 1% copper sulfate solution. Why? Because we are just dissolving 1 gram of copper sulfate into 100 ml water. Okay. So working solution, mix 100 ml of sodium carbonate solution with 1 ml sodium potassium tartrate solution and 1 ml copper sulfate solution. Then fold in Gocaltens phenol reagent, fold in Secaltens reagent, which is commercially available in a 2 normal solution. Dilute it into 1 normal before use. Principle. Once again, 2.63% sodium potassium tartrate solution, 1% copper sulfate solution, 2% sodium carbonate in 0.1 normal sodium carbonate hydroxide solution. Protein standard solution, dissolve 100 mg of bovine serum albumin or BSA in 100 ml, 0.1 normal NaOH solution, keep frozen. And working solution, mix 100 ml of sodium carbonate solution with 1 ml sodium potassium tartrate solution and add 1 ml copper sulfate solution. 100 ml of sodium carbonate solution. Sodium carbonate is acting as a primary. So 100 ml of sodium carbonate solution and 1 ml sodium potassium tartrate solution and 1 ml copper sulfate solution. Then fold in Gocaltens phenol reagent, fold in Secaltens reagent commercially available in a 2 normal solution, dilute it into 1 normal solution before use. The first step involved the formation of copper protein complex in alkaline solution. The first step involved the formation of copper protein complex. The first step involved the formation of copper protein complex in alkaline solution. The first step involved the formation of copper protein complex in alkaline solution. Amino acid like tyrosine and tryptophan in the copper protein complex then reduces phosphomolybdate and phosphotungsten present in Folin Secalter reagent to yield an intense blue color. The first step involved the formation of copper protein complex in alkaline solution. Alkaline medium will be providing copper protein complex. Amino acid like lysine and tryptophan in the copper protein complex. Copper protein complex then reduces phosphomolybdate and phosphotungsten present in Folin Secalter reagent to yield an intense blue color. Procedure preparation of calibration curve. Pipette out, induplicate into a series of dry clean test tubes 1, 2, 3, 4 of standard protein solution and make up to 100 ml with distilled water. Add 5 ml of working solution into each test tube and incubate test tubes for 30 minutes at 37 degrees Celsius. Record the intensity of color developed in colorimeter at 660 nanometer against a reagent blank. Prepare a standard graph by plotting concentration on x-axis and absorbance on y-axis. In analysis of sample, take 1 ml of given sample solution in a test tube. Take 1 ml of given sample solution in a test tube, add 5 ml of working solution and incubate at 37 degrees Celsius for 10 minutes. Then add 0.5 ml of Folin Ceocaltins reagent, mix well and incubate at 37 degrees Celsius for 30 minutes. Measure optical density OD at 660 nanometer against a reagent blank. The amount of protein present in the given sample can be calculated using the following formula. Concentration of sample is equal to absorbance of sample by absorbance of standard into concentration of standard. Absorbance of sample concentration of sample is equal to absorbance of sample by absorbance of standard into concentration of standard. Concentration of sample is equal to absorbance of sample by absorbance of standard into concentration of standard. Result: the amount of protein present in sample A from calculation from graph, the amount of protein present in sample B from calculation from graph. Here you could see a picture which depicts the observations. Concentration 0.2, 0.4, 0.6, 0.8. OD, optical densities are also shown, corresponding optical densities. Then calculations. Calculation of concentration of standard 1 mg per ml. Absorbance of standard 0.39. Calculations: concentration of standard 1 mg per ml. Absorbance of standard 0.39. Absorbance of unknown sample A 0.12. Concentration of unknown sample A is equal to absorbance of sample by absorbance of standard into concentration of standard. Once again, concentration of unknown sample is equal to absorbance of that sample by absorbance of standard into concentration of standard. Here we will be using any one of these one as standard. Okay. Concentration of standard in the sense 1. Estimation of protein, Lowry's method. Scale x-axis, I need to write that. Y-axis similarly. Concentration, x-axis, y-axis. OD at the given nanometer. Next experiment, estimation of RNA by Osinol method. Estimation of protein by Lowry's method we just learned. Next, estimation of RNA by Osinol method. To determine the amount of RNA in a given sample, materials required: test tubes, pipettes, colorimeter, Osinol reagent. Acid hydrolysis of RNA reduces ribose, which is in the presence of strong acid, is hydrolyzed to yield furfural. Acid hydrolysis of RNA reduces ribose, which is in presence of strong acid, is hydrolyzed to yield furfural. Osinol reagent with furfural in presence of ferric chloride is given green color. Purine nucleotide are generally more reactive than pyrimidine nucleotides. Estimation of RNA by Osinol method to estimate the amount of RNA in a given sample. Test tube, pipette, colorimeter, Osinol reagent, etc. Acid hydrolysis of RNA reduces ribose, which is in presence of strong acid, is hydrolyzed to yield furfural. Osinol reagent with furfural in presence of ferric chloride or FeCl3 is give a green color. Purine nucleotides are generally more reactive than pyrimidine nucleotides. Reagents: standard RNA solution, dissolve 10 mg of pure reagent RNA in 100 ml distilled water to obtain a solution of 100 μg of RNA per ml. Keep frozen. An Osinol reagent: dissolve 300 mg of Osinol in 5 ml of ethyl alcohol. Dissolve 300 mg of Osinol in 5 ml. Dissolve 300 mg of Osinol in 5 ml of ethyl alcohol. Add 3.5 ml of this solution just before use to standard RNA solution. Dissolve 10 mg of pure reagent RNA in 100 ml distilled water to obtain a solution of 100 μg of RNA per ml. Keep frozen. Osinol reagent: dissolve 300 mg of Osinol in 5 ml of ethyl alcohol. Osinol reagent: dissolve 300 mg of Osinol in 5 ml of ethyl alcohol. Add 3.5 ml of this solution just before to use 100 ml of 0.1 normal solution of FeCl3 in concentrated HCl. Then preparation of standard graph. Pipette and duplicate into a series of clean test tubes 0.5, 1, 1.5, 2.0 ml of standard RNA solution into and make up to 2 ml with distilled water. Okay. Then add 3 ml of Osinol reagent to each test tube and mix well. Keep the test tube in a boiling water bath to each test tube for 15 minutes. After 15 minutes, remove test tubes from water bath and cool to normal temperature. And measure the color developed in a colorimeter at 665 nm against a reagent blank. Prepare a standard graph by plotting concentration on x-axis and absorbance in y-axis. Here we could see results. Volume of RNA solution in ml. Concentration of RNA μg per ml. Absorbance. Calculation. Absorbance of standard 0.22. Concentration of standard 50 μg per ml. Absorbance of sample 0.23. So concentration of sample, absorbance of sample by absorbance of standard into concentration of standard. You could see the result. Analysis of sample. Take 2 ml of RNA solution in a test tube and add 3 ml of Osinol reagent. Mix well. Keep the test tube in a boiling water bath for 15 minutes. Cool and measure the OD at 665 nm against a reagent blank. The amount of RNA in a sample can be calculated using the following formula. Concentration of sample is equal to absorbance of sample by absorbance of standard into concentration of standard. Result: amount of RNA present in a given sample from calculations and from graph. Graph showing optical density of RNA. Concentration of RNA. Then estimation of hemoglobin in blood. Hemoglobin is a compound composed of protein material globin and a coloring material hemochromage. The latter contains iron and is rapidly transferred into hematine in presence of oxygen. The matter later contains iron and is rapidly transferred into hematine in presence of oxygen. Total content amount of hemoglobin in blood varies with age and sex. In adults, ranges from 15 to 17 mg in 100 ml of blood, and increase in hemoglobin is noted in condition where the number of RBC is increased. It may be caused by the concentration of blood due to less or large quantities of water, as in cholera, or due to large quantities of increase in RBC at high altitudes. Decrease of hemoglobin is a condition observed in all cases of anemia. So the principle involved in conversion of hemoglobin to acid hematine by reacting with HCl. The brown colored hematine produced is diluted till the color matches with that of standard acid hematine, kept in tubes on either side of central tube of Saheli's apparatus. From the dilution required and hemoglobin content is estimated. What is happening here? Here, conversion of hemoglobin to acid hematine. Materials required: Saheli's HB meter, hydrochloric acid, lancet glass rod, distilled water. So usually hemoglobin estimation is done by hemoglobinometer. This consists of two similar tubes on a black stand with a white background. The two tubes contain acid hematine and are sealed between the tubes. There is another large graduation tube which is open at one end. Two scales are marked on the tube, one showing grams for 100 ml of blood and the other showing the percentage of hemoglobin with reference to normal blood. The forms scale is marked from 2 to 22 and later from 10 to 140. Two scales are marked on the tube, one showing grams or 100 ml of blood and the other showing the percentage of hemoglobin with reference to normal blood. The form scale is marked from 2 to 22 and later from 10 to 140. A pipette of 20 mm capacity is also provided to drop blood. So decinormal HCl is 1.1 normal HCl is prepared by adding 1 cc of concentrated HCl to 99 cc of diluted water. The graduated tube is filled up to 10 in percentage scale with decinormal HCl. The fingertip is pricked after cleaning the tip of the finger with alcohol. The first drop of blood is discarded. The blood flow is enhanced by massaging the fingertip. As soon as enough blood is formed at the fingertip, the capillary tube of the pipette filled up with blood by sucking or from rubber tube of the pipette. The blood was being transferred into a graduated tube containing HCl. Convenient method is to carry the tip of the pipette to the bottom of the tube and to blow out the content gently. With the same pipette, sucking and blowing was repeated and the tube was thoroughly mixed. Fluid was thoroughly mixed. After 3 to 5 minutes, the mixture was diluted with distilled water dropwise until the color matches with the standard finally, and level distilled water and distilled fluid was read. Result amount of hemoglobin present: 14.5 gram of per 100 ml of blood.